What are the proteins that are comprised in our human genome? How do the proteomics work? How do all the different proteins that are in the genome, how do they all work together? So there are various techniques that we can employ to figure out what is happening at the cellular level, or be it at the tissue level. One of them is phase contrast microscopy. The other one is, say, differential interferometric contrast. So these two techniques are more morphological. So the way that we're imaging in this is that you're looking at different sort of how the morphology within a cell changes. So I should just mention that actually we're looking at HeLa cells here. HeLa cells is a very common cancer cell line. But then there are also techniques that allow you to figure out what's happening at the compositional level, and that's fluorescence microscopy, for example. So fluorescence microscopy is huge. It's one of the biggest imaging techniques that we have. And lastly, we have another imaging technique called vibrational spectroscopy, or microspectroscopy, I should say. And this is where you're actually looking at, the actual composition of a cell and say where different bits of the cell are, where's my cytoplasm, where's my nucleus, how do they change, and all of that. So the main difference between, say, the top two and the bottom two is morphological versus compositional. So moving on, so let's talk a little bit about fluorescence microscopy. It's one of the most widely used, actually, imaging methods, and what we do in fluorescence microscopy is that we take, say, imaging agents. So you need to use imaging agents with fluorescence microscopy, okay? So what I mean by imaging agents is that we've either got probes or labels that we can attach to whatever it is that we're interested in looking at. So what these probes or labels do is they allow us to either visualize or characterize or measure biological processes within living systems, because they have responses to what we're doing to the system. So if we say probe the system with a particular set of probes, then the responses that we get allow us to figure out what's going on in the cells. There are different kinds of labels that we can use. I mean, the field is absolutely vast. You've got genetically encoded fluorescent proteins, so GFP, which is something that maybe all of you might have heard about. It was sort of the breakthrough in fluorescence microscopy. You've got organic dyes, so the manufacture and the synthesis of organic dyes is a very big field. You can have loads of other different things, that basically the main characteristic being that you have to have them be fluorescent. And so one of the things I was going to talk to you about is actually luminescent organic nanoparticles, which is something that we're doing at King's. So here's a little image, a video of the nanoparticles under a camera. So these are being imaged onto a CMOS camera. And in the bottom image, you actually see that when irradiated with a source of light, you can see that they're all fluorescent in different wavelengths. So you've got blue, yellow, orange, and red. And on the top, you can see that all of them have been taken up by HeLa cells. So you've got a range of wavelengths. So this is actually non-specific binding. So you just take your HeLa cells and you get them to take up these nanoparticles, and then they glow, to use the term sort of crudely, is that they glow under different conditions, and you can see where all of them are placed. So you have variation here in terms of what kind of particles can you see, where can you see them, and different colors. So you can actually supposedly even see 4 different areas if you wanted to in here. So this is another image. So on the left hand, you've got, say you've got your bright field of your cells, and on the right hand side, you've got the take-up of the nanoparticles within the cells, visualized under fluorescence microscopy. And lastly, the image that we have at the bottom is when you're doing something called two-color confocal microscopy, in which you've, say, taken 2 labels of interest, gotten them to go to 2 different places of interest within a cell, and then you map that in your fluorescence microscopy to say, right, this is where my nucleus is, this is where the rest of my cell is. Now I can see processes or events or intercellular, mechanisms, whatever it is that I'm interested in. So this is a little video of when you've gotten your nanoparticles to be taken up by your cell. You can see they're sort of gathered around the nucleus, and it's been superimposed onto the bright field image of a cell. So you can see the rest of the cell as is, and your nanoparticles, fluorescent nanoparticles, in a particular area of interest. So moving on. So this is something that I do more on a regular basis, which is by Raman spectroscopy. So Raman spectroscopy is another kind of really strong imaging technique that's emerged over the past sort of 2 decades or so. It's been around for chemists for a really long time. It's an analytical technique. But what it basically does is it probes It probes whatever it is that you're looking at, and the response that you get from that system is chemical-based. So it really is a representation of all the chemical bonds that you have within your system. So say, for example, my OH bonds would have, respond in a certain way and give me a certain signal. My CH bonds would respond in a different way, give me a different signal. So these differences of signals, which we call differences in energy levels, is what allows you to map the composition of an entire analyte, or whatever it is that your specimen that you're interested in looking at. So this is a really strong compositional technique because it allows you to set up a really strong biochemical, image of your samples, like, where are my lipids, where are my mitochondria, for example, where's the ribosomes, where's the nucleus, because everything has a different bond structure, and because we can associate energies to different bonds, we can actually create something like so if we move on to the next slide, we can create a barcode, so to speak. So this sort of signal that you're seeing is representative of a certain type of cell. So all the energies that are represented here are representative of different bits of my cell. So for example, I can be looking at proteins and I know they'd be in, say, range X1 to X2. I can be looking at lipids and they'd be in range Y1 to Y2. And these are all things that are determined. So you can have like a really nice barcode that you can establish. So the reason that we use this is, say, we want to do the chemical mapping of proteins. We have two different kinds of proteins. They'd be ever so slightly different because their composition would be ever so slightly different. And if I want to compare the two, their barcodes would be ever so slightly different. And that difference in their barcodes is what allows me to figure out what's happening within their interactions. So if I move on to my next slide, so you see over here, you might just be able to see that, for example, I can see where my proteins are. So a carbon-carbon bond, for example, that's where my protein section is. And I can see sort of around the 700 to 800 region, I can see my DNA, RNA signature. If you move further all the way to the end, you can see some more DNA structures. In between, you have carbon-hydrogen bonds. You can see where my lipids are. So it's all these little areas that are well-defined, and changes within those areas would reflect changes in the barcode, and then you can figure out what's going on from there. So how do we use this? So for example, if I talk about compositional analysis, right? I take a cell, I don't really know what I'm looking at, I run it through my spectroscopy system, and I run it at a particular signal, say for example, right? I decide I'm going to choose X. I generally know that around X, I might be expecting, say, the cytoplasm, and if I'm around about in the right zone, then I can figure out that, okay, around 750, this is what my cytoplasm looks like. At around, say, what am I looking at, 1689, that's where I can expect my proteins to be. At around 2855, around that region, that's where I can expect my lipids to be. If I put all of them together, then I can get a really nice image of what my cell looks like, and where all the sort of the components of the cell, how they're distributed within the environment of the cell. So that's what I mean by compositional analysis. What else? So what does this allow me to do? It allows me to look at different things that are happening within the cell. So over here, for example, you've got images of the division of chromosomes, or you've got images of sort of the last phases of cell division. What else does it allow me to do? It allows me to look at a 24-hour cycle, for example. So I can look at different parts of the nucleus of my cell, see how they progress over a certain time period. So you can do time-lapsed measurements. What about tissues? We can also, of course, look at tissues, and you can map a normal tissue region. So this is the brain tissue. You can map different areas of the brain because they all have different compositions. But what you can also then go on to do, and this is Raman spectroscopy, something that's being used quite commonly in histopathology, is that you can establish what a healthy tissue looks like. So for example, in cancer diagnostics. And then you can also establish what perhaps a diseased one would look like. So if I go to my last slide in diagnostics, so you have on one side a solid tumor, and on the other side you have a tumor margin. Because previously I've established what my normal tissue looks like, and then I can go on and say, oh right, this little bit of the tissue that I'm imaging actually looks quite different from my normal tissue. So that means that's where my tumor must be. So you can either look at the whole tumor region, or you can look at a tumor margin region and then allow histopathologists to figure out, right, this is all the cancerous region, this is probably what we need to get rid of, this is healthy tissue, this is what we can keep. So one of the things that Raman spectroscopy is actually doing is allowing histopathologists to run this sort of system on algorithms. So you're taking sort of a slightly human element out of it, because the algorithm then is a machine-learned algorithm. So what it does is that it'll just keep running it and figure out where my diseased tissue is and where my healthy tissue is, and hopefully allow you to figure out which part of the cell is healthy, which part of the tissue is healthy, and which part's diseased. So that was a little brief, albeit quite fast overview of bioimaging. So what we've learned is that, if I move on to my conclusion, is that you can have imaging of a particular area, and it's quite integral to learning how biological processes interact, how proteomics work, how amino acids interact with each other. We're quite integral to the field of life sciences. And many of the cellular structures that we have, they're sort of, you can look at intracellular events, you can look at interactions between different parts of the cells, and it's allowed us to, gain knowledge of biological processes at that microscopic level. So, and what can we see? Well, we've been able to look at different kinds of techniques, and they allow you to have either a morphological characterization, a compositional characterization, look at time-lapse events, look at interactions, look at specific or non-specific imaging, and finally be able to look at diagnostics and be able to model a system on the basis of that.